بسم الله الرحمن الرحيم. النهاردة إن شاء الله هنتكلم على الانتجومنتري سيستم. The integumentary system. integument يعني غطاء، وطبعا الغطاء الجسم هو الجلد or the skin. So the integumentary system is the system that covers the body, and it is formed of the skin or cutaneous membrane and the accessory structures. The skin or cutaneous membrane is continuous with all the openings of the body, such as the mouth, the nostrils, the eye, and the external ear. The accessory structures are the hair, the sebaceous and sweat glands, and the nail. The skin or cutaneous membrane is the largest system in the body. It is about 2 square meters and it weighs at least 4.5 kilograms. The functions of the skin. The skin protects the body from bacterial invasion. It also protects the body from drying out. It absorbs ultraviolet radiation by its content of black pigment, which is melanin. It regulates the body temperature, as we will see. It provides sensory reception. It synthesizes vitamin D by the help of ultraviolet rays of the sun. It serves as a reservoir for blood and nutrients. The layers of skin. The outermost layer covering the skin is called the epidermis. Underneath the epidermis is the dermis. The epidermis is formed of stratified squamous epithelium, as we will know, and the dermis is formed mainly of connective tissues. The dermis has 2 regions: the papillary layer underneath the epidermis, and this is formed of loose connective tissue, and the reticular layer, which is formed of collagenous connective tissue. Under the dermis is the hypodermis, which is formed mainly of fat cells, and it binds the skin to the underlying structures such as the muscles. Types of skin. There are 2 types of skin in the human body: thick skin, which is found on the palms of the hands and the soles of the feet, and thin skin, which is found all over the rest of the body. The thick skin is called thick because the outermost layer, which is the epidermis, and the keratinized layer are extremely thick, especially the keratinized layer, while the thin skin has reduced stratum corneum or keratinized layer. However, it contains hair and sebaceous glands. That's why the thin skin is also called hairy skin. The epidermis. The epidermis is the main function of the epidermis is to protect the body from harmful influences from the environment and against fluid loss. The epidermis is formed of stratified squamous keratinized epithelium, and as you know, such epithelium do not have blood vessels in between cells, and so it is nourished from the underneath layer, the dermis, by blood vessels of the dermis. However, it contains naked nerve endings between its cells, and it acts primarily in pain sensation. The epidermal cells. Most of the cells of the epidermis are keratinocytes. Keratinocytes because they contain intermediate keratin filaments in the lower cells, and it contains keratin, soft keratin, in the upper layers. The others are non-keratinocytes, which represent only 5% of the cells. The keratinocytes have keratinocyte stem cells or epidermal stem cells, which are regenerating keratinocytes, continuously dividing to regenerate all the keratinocytes of the epidermis, and they are found in the basal layer. And corneocytes on the top of epidermis, and these corneocytes are dead cells which are continuously peeled from the surface. And so in the keratinocytes we can see that they are continuously formed from the lower cells of the stem cells or the stem cells, and continuously lost from the top layer or the surface layer. The non-keratinocytes are Merkel cells, and these are touch or tactile sensing cells, and the Langerhans cells, which are cells important for immunity, and they are dendritic antigen-presenting cells. The third type are melanocytes, which synthesize melanin, the dark or black pigment, which gives the skin its color. The epidermal cells are organized vertically according to their histological features into 5 layers or strata. The dermis and epidermis are connected firmly together by the basement membrane. On the basement membrane, the first layer of cells is called stratum basale, and it is formed of columnar to high cuboidal cells. It's also called stratum germinativum because it produces new cells by division or cell proliferation from stem cells and the other basal cells. The stratum spinosum are cells which have short processes between them, and the short processes are called spines. Spines يعني شوك أو إبر. So it is like it is spinous and have spines in it, and these short processes occur because during preparation of slides, the cytoplasm of the cells in this layer shrink, and they leave finger-like processes or spines connected by desmosomes, as you see. Then, above that is the stratum granulosum. It's called granulosum because the cells have darkly stained granules within their cytoplasm. Above the stratum granulosum in the thick skin, there is a layer called the stratum lucidum, or transparent layer, and it is found only in thick skin. Above that is the stratum corneum, which is formed of cornified dead keratinocytes, and they have cells called corneocytes, or dead cells called corneocytes, which shed from the top all the time. And so we will see that the epidermis is continuously renewed by the formation of cells in the basal layer and the loss of cells on the top layer. This is called epidermal homeostasis. So the skin epidermis undergoes continuous renewal every 3 to 4 weeks by a process called homeostasis. Stem cells in the basal layer of the epidermis provide new cells to replace those that are constantly lost by shedding from the surface of the skin. How does this occur? We'll talk about the morphological and biochemical changes during keratinocyte cornification and apoptosis. We said that the first layer of keratinocytes rests on basement membrane, and they contain the stem cells. The stem cells divide in order to give a stem cell and transit amplifying cells. Both of them are attached to the basement membrane, and so the division is a horizontal division. The transit amplifying cell can divide 3 to 4 times horizontally to give cells also attached to the basement membrane, or to the basement membrane or basal lamina. These transit amplifying cells, after 3 or 4 times of division, start to divide vertically, and this vertical division results in the formation of differentiated parabasal spinous cell, which is not connected to the basement membrane or basal lamina, and another one which is still connected to the basement membrane. The upper parabasal spinous cell is a differentiated cell. That's why this is called the stage of differentiation. These cells are differentiated and cannot usually divide. Usually they do not divide, but they are very active, biologically active, in order to form the upper layers of cells. The parabasal cells are characterized by having desmosomes and keratin intermediate filaments. When new parabasal cell is formed, the older one is shifted to above the newly formed parabasal cell, And this cell is granular keratinocyte, called granular keratinocyte, and this starts what is called terminal differentiation. These cells, which are granular keratinocytes, first of all, are attached to the neighboring cells by tight junctions. As you know, tight junctions do not allow anything to pass between cells. Everything has to pass through cells only. And so it prevents the dehydration of skin by preventing water from passing from the dermis to the outside environment. The granular keratinocytes are called granular because it contains granules, darkly stained granules called keratohyalin granules. The keratohyalin granules are enzymes and special proteins that are capable of cross-linking the intermediate filaments and of the desmosomes and the intermediate keratin filaments in order to form soft keratin. These cells also synthesize structures which are formed of lipids and carbohydrates and called lamellated bodies. These lamellated bodies move to seal in between the cells, and so they are found in the intercellular spaces, and they seal the cells and prevent the passage of water or any substance dissolved in water from the surface to the basal part or vice versa. When the granular keratinocytes form that, the parabasal spinous cells, the new parabasal spinous cells, pushes the keratinocyte to the upper level, and the cells now start a process called cornification. The cornification of keratinocytes starts with the formation of a cornified envelope under the cell membrane of the cells. The lysosomal enzymes start digesting the nucleus and the cellular organoids within these cells, and so this is a process, a special process of apoptosis that occurs in epidermis. The resulting cell is not actually an active cell, but it is a corneocyte which is keratinized corneocyte. The in between the corneocytes and the underlying cells, kallikrein enzyme, which is formed by the lysosomes, dissolves the intercellular substance and gets the corneocyte free, and desquamation occurs. So, one stem cell can divide to give 9 TA cells, and these TA cells can differentiate into spinous cells and the granular cells and keratinocytes. The cells that develop from one stem cell and covered by a hexagonal corneocyte, these cells are hypothesized as the epidermal proliferative unit. The epidermal proliferative units could be identified by a special histochemical staining, by lineage tracing analysis, and this provided evidence for the contribution of epidermal stem cell during normal homeostasis to form the unit which is called the epidermal proliferating unit. So, the skin surface is covered by several epidermal proliferating units. Each of them is formed from one stem cell. Of course, any abnormality that could be found in the stem cell will result in abnormal formation of epidermal unit, and this will appear as abnormal structure. Non-keratinocytes. The non-keratinocytes will start with melanocytes. Melanocytes are melanin pigment synthesizing cells derived from the neural crest. They are located among the cells of the stratum basale. The melanocytes are round to columnar cells. Their long processes penetrate the intercellular spaces of the stratum spinosum and become connected or become in between the cells in order to send its contents of pigment to these cells. How does this occur? The melanocytes have dendrites which are in contact with keratinocytes. They send their melanosomes into the dendrites, and the keratinocytes phagocytose these melanosomes to form the skin color. Usually the melanosomes are aggregated at the supranuclear region, which is facing the sun rays and the ultraviolet, and so the pigment prevents the effect of the ultraviolet on the nuclei of keratinocytes. At the upper part, in the granulocytes, the lysosomal enzymes digest the melanosomes, and that's why the peeled corneocytes from the surface do not have a color, because the lysosomes digested the melanosomes. Merkel cells. Merkel cells are epidermal fine touch receptors. They are especially abundant in the finger tips and the oral mucosa. Merkel cells are neuroendocrine cells in the intercellular epidermis of human skin. Interfollicular means the epidermis, which is found between the hair follicles. Usually it's found as single cells parallel to the basal lamina contained among basal keratinocytes. Together with a surrounding group of keratinocytes, they form specialized epithelial structures termed touch dome. They develop from epidermal progenitors in the touch dome. Innervation of Merkel cell. The Merkel cells are found in the basal layer of epidermis. They are connected to myelinated sensory nerve that traverses the basal lamina to approximate the Merkel cells. Each Merkel cell and the nerve ending form what is known as the Merkel cell neurite complex. And so we have the Merkel cells and the nerve terminals which form the neurite complex. Langerhans cells. They are dendritic antigen-presenting cells. Dendritic, that is, they have processes in different directions. They ultrastructurally, they have an indented nucleus, and the cytoplasm is electron-lucent cytoplasm. Tight junctions are found between them and the keratinocytes. That's why the microorganisms and foreign antigens found on the surface of the skin cannot pass between the cells, and they have to pass through the Langerhans cell. In this case, they activate Langerhans cells. How? Environmental microorganisms and antigens, as we said, cannot pass through the cells because of the tight junction. And so they have to pass through the dendrites of the Langerhans cell. This activates the Langerhans cells, and as it is activated, it will move down to the dermis, where it will get into the lymph vessel towards the lymph node. They are replaced by dermal Langerhans cells from the dermis.